And this is Sue. Welcome to
Front Porch AF,
where we, here, let me consult in my notes. What is it we're doing? Okay, we're going to gather and have a drink and a laugh and tell them what else we're going to do.
We're going to solve all the world's problems.
We are solving the world's problems today, especially on today's show. Ooh, but we'll save that for later.
Yes.
Okay, how have you been?
Pretty good.
And
Just
you
got
flew in.
Yeah, just got off an airplane this morning.
And boy, are my arms tired. You flew in from a coast.
From the West Coast. Yeah, I was out in
Coast.
the sunny West Coast.
Did you sleep on the plane at all?
Yeah, the first flight I slept pretty good.
You had to change planes?
Yeah, absolutely.
When?
In the middle of at 6 a.m.
You mean you didn't fly first class directly from?
No.
Oh.
No, sorry.
You had to change
I did
planes.
get upgraded to first class on the way back in Charlotte when I flew from Charlotte.
Oh, nice.
Yeah.
What was for breakfast?
You know, I don't like plain food, so nothing.
Oh, darn. I love any institutional food. If it comes in little packets and it has a place for everything and everything in its place.
No, I'm getting all icky. Let's talk
I
about
actually
something else.
asked to stay overnight in the hospital for my hip replacement so that I could get hospital food.
You are weird.
I am. Hey, welcome back, everybody, to our fourth, count 'em, 4
That's right.
episodes. Speaking of our third episode, we have some feedback.
Oh, let's hear it.
Okay. Feedback. The last show, which was communicating with lawmakers with our special guest, Tanya Hancock, our Metro Council member. One of our listeners, Janet, wrote in and said she wished her council member was as engaged as Tanya. So there you go.
Yeah, Tanya's awesome.
What else? We had a nice note about music, about the lead-in music.
Oh, yeah.
And guess what I did this morning? I put the album up on the website.
Oh, yeah.
So that people can go listen to everything from Chug It Down and Go.
Yeah, listen to that. That's good stuff.
Our bar friends. So I walked into Dee's last night. Like 7 people came over to me and said, I listened to the podcast. Most of them said it's great.
Mm-hmm.
The one who didn't was stuffing a snack food in his mouth.
So he just didn't say anything?
He didn't say anything.
Well, that was polite.
Yeah. Hey, what have you been hearing?
Oh, I've gotten great feedback. You know, the consistent thing I've heard is I feel like I'm sitting right there with you.
Well, scoot over and ask them what they want to drink.
All right. What are you drinking today, by the way?
Oh, drink of the day. You're going to love this.
This is different.
This is my Grandma Linnie's Cocktail. When I was about 6 or 7, I used to have to grandma sit. When my parents would go out on Saturday night, they would take me over to my Grandma Linnie's house where we would watch Lawrence Welk, and she would have her single glass of Mogan David wine.
Oh, my grandfather drank Mogan.
Oh, I know. I thought it was really good. And she would make me a jelly glass with like a teaspoon of Mogan David and the rest filled with ginger ale.
This is where it started, folks.
Uh-huh. And this is, by the way, there's more than a teaspoon of wine in this one. What are you drinking? No, but I'm sorry.
Yeah.
Go ahead.
I'm drinking Miller Lite, as usual.
Yes,
yeah, but the huggy.
It's not a huggy. It's a koozie.
The huggy for the episode is LSU Tigers because it's March Madness and the LSU women's basketball team is in the Elite 8 and they play tomorrow. So this is
all right.
You know,
I'll watch tomorrow. Guess what? I guess Tennessee won.
I don't know.
By the time this airs, all of this will be old news.
Yeah.
So
the women are playing this afternoon.
Oh, well,
they play Texas.
Did the men go and are they playing some? Anyway, it's a thing with a round ball and they go through a hoop and
and guys run up. I don't know, but the women will play today.
Okay, Neighborhood Notes is next on our so important list. Go ahead.
What was going on while I was out of town this week?
Oh my God. Okay, on our Facebook page. All right, The Bend has a Facebook page. Earlier this week, it was posted. 3 young calves have jumped the fence and are somewhere off of Bend Road. 2 black and 1 red. Please call 615 blank, 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 blank if you see them. Now,
well, what happened?
Mark, wait a minute. We have an update.
The cows were recovered. The cow was, but there's an update this morning. A black cow looking bewildered outside the fence at 1900 block. And then there is a comment that says, sending owner a message.
That means somebody actually knows the bewildered cow.
Yes.
The bewildered cow will be rescued. I'm positive. It seems like that help is on the way.
Well, and we can always go down the ATV and see if she's still there.
That's right, because the picture that we're going to post for this episode will be Kelli in her ATV with our special guest, but more anon. By the way, have you ever heard of the word anon? A-N-O-N?
I mean, like anonymous? Like
No,
QAnon?
anon. Like Shakespeare. More anon. Coming soon.
Okay.
You know, it would be like if you had a movie poster.
Yeah.
And instead of coming soon, it said anon at the top.
Okay.
Perhaps I oversimplify things. Tell me, okay, LSU, neighborhood notes, drink specials, feedback. Now, we are so excited. I've got to tell them about this one.
Oh, yeah, you do.
This is way over. I am over the moon. Kelli is over the moon. We are all over the moon. Go ahead, tell them about one of the inspirations for our podcast.
Well, one of the inspirations for our podcast is the Bulwark
Yes.
and everything that they do because they're awesome. And Sue,
we got a note.
Sent a lovely email, by the way.
Yes.
Was just really well crafted.
We from one of the main guys or girls at the Bulwark saying that they had signed up and they had shared. I'm blushing here. Can you see this? They had shared the podcast with the team. So yes. So and thank you.
It's pretty amazing.
Yeah. And keep those cards and letters coming, folks. We really.
Yeah, we love hearing from everybody. And it helps us out so we know we're doing what you guys want to hear.
And we take your suggestions really seriously. Matter of fact, we're changing the location for one of our upcoming shows, which I'll tell you about later, because of the suggestion of one of our listeners. I keep saying readers. I was in publishing for many decades, but it's truly listeners.
And what was the suggestion?
The suggestion was, I've got to save it till the end because that would give away the show.
Okay.
It's the locale. Instead of a front porch someplace, it's going to be a front porch another place that has some animals
traveling
on it.
Front porch.
A traveling front porch.
I love
the whole
it.
Concept. Okay, but now I've got to turn it over to you because, ladies and gentlemen out there in podcast land, the last biology course I took was in 1963. I bet you the science has changed just a little bit. So I'm going to turn it over to my science partner.
Okay, today I'm the science partner. And we're super excited about the special guest we have with us today. And this stemmed out of some news in the headlines recently about mice.
Transgender mice.
Not exactly.
Those Mickey or Minnie. What is it?
It could be either.
It could.
It could. But we want to kind of clear this up for folks and bring it down and help people understand what actually it means when we talk about genetically engineered mice that are transgenic mice instead of transgender. So we have some eminent scientists with us today who've been working in this field forever and know a lot about these mice and about biomedical research. We're really happy to have with us today Dr. Jeanne Wallace. And Jeanne was born in Kansas, grew up on a small farm, and she stands straight. Like, have you ever been out there and all that wind? Yes. And I wonder how people can not grow up walking sideways, but she's tall and looks great. She did her residency and fellowship after she did Doctor of Veterinary Medicine at Kansas State University and then went on to Wake Forest University Medical Center to do her residency and fellowship. And then for the last 40 years, she's been leading animal care and use programs and leading clinical care of research animals, which we're going to be talking about today, in large academic medical centers where a lot of this research happens that helps us and changes the way that we do medicine and finds cures for people. Mm-hmm. And in addition to Jeanne, we're really honored to have Dr. Ron Emeson with us today. Ron is a distinguished professor in pharmacology, neuroscience, and molecular biology. And he started out in Denver, Colorado, which I didn't know that about him until I read his bio. And then he skipped all over the country. He was at Johns Hopkins where he did his Ph.D. And then...
So the law was after him?
No.
No. Okay. I'm totally misunderstanding the word skip.
He wasn't evading the law. Okay. And I mean, he got his BS at Hopkins and then went on to University of Colorado Health Sciences Center and got a Ph.D. Then went to UC San Diego and did postdoc. And now he's an endowed professor. And he has his own research lab, and he studies how our genetic code affects our brain and how that affects our metabolism. And he also, as part of this and part of his job at one point, he was in charge of the center that created these genetically engineered mice, these transgenic mice. So he's like a super expert in this, and he's going to be able to break it down so we can all understand it.
I cannot believe. Thank you so much for coming with. Now I'm going to be learning something.
Yeah.
So welcome to our front porch.
Yeah.
Dr. Jeanne, Dr. Ron.
And so Jeanne and Ron, introduce yourselves to us.
Well, you did a pretty good job already. I guess the only thing I would add is, yes, I was born in Kansas, but I was born an introvert. And so this is really pushing me outside my comfort zone. I'm just going to say that right up front.
We're not in Kansas anymore,
No, we're
Toto.
not in Kansas anymore, are we? And I guess it actually is probably a good point to make is I think that one of the things that we as scientists have not always been good about is talking about what we do. And it's not because we were trying to hide anything, but because we're generally nerdy and sort of introverted. I assure you that Ron does not have a white coat on and he's not in his basement working every day. He does some really important stuff. So we appreciate the opportunity to be here and share what we do with everybody. So even though it does take us a little bit outside of our comfort zone. So,
yeah. Ron?
Well, it's a pleasure to be here. And I guess going off of what Jeanne said, there was a rally for science at the Capitol here in Nashville a couple weeks ago.
Yeah.
And there was a sign that kind of stuck with me that said, things must really be bad for science if introverts had to come out.
Yeah, that's so true.
It is.
And I think that's for most scientists, we are perceived as nerdy and we're just focusing on our work. And, we don't interact with other people maybe as well as we could. And so maybe our communication of what we're doing to the general public doesn't come across as well as it might.
By the way, just may I insert something?
Yeah.
I have understood every word so far. So keep up the good work, everybody. All right, now I'm going back.
I do have to tell one short story then.
Okay.
When I got my Ph.D., I had my dissertation and I had a bound copy that I gave to my parents and they put it on the coffee table and they were all proud of it. And 10 years later, it was still sitting on the coffee table. And I asked my dad if he'd ever looked at it. He goes, "Oh, yeah, I read some of it." And I said, "Oh, what did you read?" He goes, "Well, I read the abstract, which, a couple paragraphs in the front." I said, "Oh, did you understand it?" He said, "I understood a lot of it. I understood the, a, of, because."
Oh yeah, I think it is.
We'll try to do better today.
Oh, that's what, and I know you guys well, and I know you can do that, and I think that's also part of the communication. Is when you're in science, you develop this language that is not this. You say things that you never say in a normal conversation. Yeah. So, and that kind of feeds into what I would like to do next. And Ron, this one is on for you, and it's because you have this expertise. But can you explain to our listeners what a transgenic or genetically engineered mouse is in a way that we could understand it?
So one of the reasons that you're probably asking, the reason that this came up is there's been some confusion in the press recently about transgenic versus transgender mice. And let me say at the offset that there's really not any work going on in transgender mice. That's not really a thing. I could imagine that there is a lab somewhere that's doing something related to that, but that's not really a thing. Mm-hmm. So transgenic mice was a term that was developed in the late 1970s, early 1980s. And what that is we develop the ability to introduce DNA from from one organism into another organism. So we started out, scientists like to grow cells in a dish in culture, and we can introduce genes into those cells. And people wanted to know whether or not we could do the same thing with a whole organism. Yeah. And so mice are a good example, are a good way to do that. And so in 1982, the first transgenic mouse that I'm aware of was developed by two investigators, one at the University of Washington, one at Penn. And what they did is they took the human growth hormone gene. So this is a gene that encodes growth hormone, which is hormone that's made by your pituitary gland at the base of your brain, and it causes all the growth that you do during your life is promoted by growth hormone. They took the human growth hormone gene and they put that into a mouse. Okay, so you go, well, why did they do that? And so you might be able to make predictions. What would happen if a mouse had too much growth hormone? And I would say from the outset, not knowing anything else, I'd expect the mouse to grow, maybe larger than a normal mouse.
Was that the case?
So it turns out that was exactly right.
Yeah.
So normal adult mice weigh about 35 grams, and these mice weighed about 85 or 90 grams. So 2.5, 3 times as much as a normal mouse.
Have you ever seen that movie Attack of the Lagomorphs?
No. Is it anything like Attack of the Killer Tomatoes?
Well, it's those giant rabbits, right? These rabbits are like car size. Anyway, that just popped in.
So those are called transgenic animals. They have a trans gene that came from another organism and put into that
and
into
for
their
all of
genome.
You, for all of you who didn't take Latin for 3 years back in 1960, trans means a cross. You're crossing. You're going from one thing to another. That's it for me.
Yeah. But Ron, I mean, probably the majority of the mice we work with now are not necessarily transgenic.
They're genetically modified.
Right.
So even scientists get that confused. Because transgenic mice were the first to be developed, they just refer to all genetically modified mice as transgenic mice, but that's not true. So transgenic mice, as I said, you put in another gene in trans from another species. But genetically modified mice, now we have tools. I think probably a lot of people have heard in the press this technique called CRISPR.
Yes.
Which allows you to modify individual nucleotides, individual bases within the genome. So instead of putting in the human growth hormone gene, you could take the mouse growth hormone gene, because they have one as well, and you can modify it however you want to do it.
Right. You could make it go stronger, or you could totally turn it off.
You can turn it off. You could make it more. You can amplify it so that the mice would be larger just because they're making more of mouse growth hormone, not necessarily human growth hormone.
Has this translated to humans, the transgenic studies, yet?
So there are beginning. I'll answer your question, but I kind of want to come back to it.
Oh, okay.
Yeah,
I'm sorry.
Let's come back to
All
that.
right.
Let's move on.
I'll put a pin in
Put a pin in that, because what I would like for Ron to talk about is, like, why would we do that? Like, how, as a scientist in your lab, I know you work with these types of animals. Why would you do something like that? Or how does it, give us an example, like a real world example of how this happens?
Okay, I'll give you a real world example, but I'll preface that by saying we're talking about mice now.
Mm-hmm.
What I'm saying isn't specific for mice. There are lots of different kinds of organisms that are used in biomedical research. Mice are popular because they breed quickly. They're very similar to people. They're 85% identical, the DNA between mice and humans. And if you look at the genes that mice have versus the genes that humans have, we're 99% identical. So as a model system for trying to model human diseases, they're a really good one to go to.
And we can do it in fast forward because they don't live as long, so we can.
Right. So you don't have to wait 70 years for somebody to get Parkinson's disease. If you have a gene that you want to modify that you think is associated with Parkinson's disease, mice only live to be about 2 years old. So you can get the work done a lot more quickly, because I don't think that Parkinson's patients want to wait 70 years, for the next therapeutic strategy. So we use a lot of different organisms, zebrafish, worms, all the way up to some non-human primates, because there are some kinds of things that we want to understand that you can't really model in mice. But for the most part, people are using mice. So an example would be to model a human disease. So I'll use an example from my own laboratory. There's a neurodevelopmental disorder called Rett syndrome, in which it's a rare disorder, 1 in 10,000 girls, and they learn to walk and talk, and everything is going okay till about 3 years of age, and then they lose the ability to do that. And we know the mutation in the gene that's responsible for that. So we can go into the mouse, into that gene, and make the same mutation that people have, and then ask, do the mice have Rett syndrome? And for the most part, they have many characteristics of Rett syndrome. So now if you want to develop a therapeutic strategy for Rett syndrome, you don't want to necessarily try it on people first. You can try it on mice that have what the equivalent is of this human disorder.
Perfect. Yeah, that's pretty cool. So, having those kinds of mice around requires an amazing facility, right? You have to have space to house these mice. And Jeanne, what are the things you have to think out from a veterinarian when you're supporting, like you support hundreds of scientists doing this kind of work? And so that's a lot of mice. So what do you, from a veterinary perspective, how do you think about that? Are there special things you have to consider for this kind of stuff?
Well, absolutely. And I think one of the things that bears saying is that whether you are in favor of the use of animals in research to or not, that's another whole podcast, I think, probably, right? But what people often don't know is that there are all sorts of, it's one of the most highly regulated activities at institutions. There are all sorts of requirements that are in place anytime an animal is going to be used for research. And as Ron is saying, the vast majority of the animals that are used are mice and rats. And you'd be surprised. I have some people say, you have all that for mice and rats? But yes, we do. And it's really important. We're evaluated by external agencies as well. But all of the things that you would think and more in terms of caring for your own animals, we have to think about in terms of caring for these mice. And because some of them have had or have been manipulated in a way that makes them highly susceptible to diseases or have shorter lifespans, et cetera. There are some certain extra special accommodations that need to be made, protections that are in place. So veterinary care, the animals have to be cared for every day, 24/7, 365 days a year. It's quite a lot to keep up with. And I often think to myself, the animals at work are sometimes cared for better than my own animals at home. That sometimes
right. Every animal has a veterinarian assigned to it, right?
Pretty much. Yeah, we have
and veterinary technicians,
veterinarians,
and then a whole slew of caregivers.
Yep, it takes a lot to do to support the research that's being done, and one other sort of tidbit, I think a lot of people sometimes ask me as a veterinarian, and I know my colleagues as well, is like, well, how can you support animals that are having research done to them? And I think the big thing that keeps us all here is, number one, a belief that we truly do make a difference in the animals' lives by being there, by helping ensure their welfare, and also that we truly, honestly do believe that it's the use of animals is necessary to improve in research, to improve our understanding of human and animal health. A lot of advances that come from animal research are actually help take care of or help with new treatments and advances in medicine at your private practice for your dogs and cats. And so,
that's one that we probably don't hit on enough, is that anything we discover in humans is often translated back to our veterinary species, right? And so your dog,
my dog and Mark,
I think the Ozempic
same pill.
Yeah.
And Jeanne, that's a great lead in to talking about shifting. I mean, animals are definitely, you can't study these diseases in a dish. These whole system diseases, and they're very important to biomedical research in general. And I just wondered if you guys had any examples you wanted to share or talk about that related to like where animals have this type of animal research or genetically engineered mouse research has made an advance in the biomedical research field.
Yeah.
I'm trying to pull a specific example. There are so many.
Yeah.
Almost all human diseases now that have a genetic component. There are mouse models of those diseases. And so all the work that's being done to develop therapeutic strategies is being done on these mouse models, whether it's Parkinson's disease or Alzheimer's or cancer.
Cystic
fibrosis, all of those.
Right.
And I don't know if you want to go back to it now, but people are starting in clinical trials to modify the genome of people to treat specific genetic disorders.
Right.
So if you have a mutation that's associated with Parkinson's disease, right? If you could go and change and fix that in it has to be in every single cell where that gene is expressed, or in the majority of them.
Yeah.
So there's still a bunch of issues with delivering that to all the right places, but all the tools are starting to come into place where we should be able moving forward to treat all kinds of genetic disorders.
Right.
That's what.
And Sue, I think to your point, or to your question, it kind of playing out forward from what Ron said, is like, without this animal model work, we can't figure out how to get these fix into every cell, right?
Yeah, into every cell. That's, I mean, well, how many cells are there? How many billions of cells are there in a human body? You can't go in and inject each one.
The scientific answer to that is a lot.
A lot. Which hasn't changed since 1963.
But I guess, thank you guys for laying this out, and kind of I want to shift gears to hear your perspectives on, at the federal level, there's these major cuts that are happening to research grants specifically, and to indirect cost, which I don't think a lot of people understand. And I think you probably may personally both be impacted by this. And I wanted to get your perspective of how does this affect the scientific pipeline? What will this do if these cuts go into the measure of cuts that we've been reading about actually go into play for the scientific pipeline?
You want to start?
You
I'll start. Okay. It's a critical question. There are big cuts looming to science in general, and you see in the headlines every day, Columbia, $400 million reduction. I think Vanderbilt recently had a $250 million reduction, and you can go institution by institution. As far as the pipeline, it takes a long time to become a scientist. So, in addition to going to college for 4 years, I went to graduate school for 6 years after that, and then I did a postdoctoral fellowship for 5 years after that. So, you take those, that's 15 years worth of education just so that I could have a lab and begin to do what I'm doing. You could imagine that if these cuts in science continue for an extended period of time, people that are in high school, people that are in college, they're going to start to think, there's no future in biomedical research for me. There's no money that's available. I'm going to go do something else. And what you'll end up doing is destroying, I don't want to overspeak, but basically destroying advances in biomedical research for at least a generation, because you have those people that decide not to do science. And one of the things that really stands out is, regardless of where you went to school, regardless of how smart you are, you never know where the next big idea in science is going to come from. It can come from a big lab, a little lab. And so you don't know who's going to be responsible. And if those people don't end up pursuing a career in science, you've lost a lot of cures in the future.
Or if they decide to finish their career in science in Britain or in France or in China.
Right. Actually, I was going to mention
Oh,
that
good.
in terms
Good.
of
Tell
the
me. Yes,
shorter
talk
term,
to that.
one of the shorter term effects. Ron has really spoken, I think, eloquently to the long-term effects, just in terms of the pipeline coming down. But the more immediate effects, I think, are, these reductions are going to result in people having to be let go. And I know there's specifically conversations about taking fewer graduate students. There's a position after graduate school where people do postdoctoral fellowships. Those postdocs are the engines behind getting the research done. As we kind of shrink, the reduced funds shrink the number of those positions, the work that is even funded right now will not be able to be done. And I have heard as recently as yesterday that Europe is going, okay, the U.S. is imploding in terms of its research engine. It's been the preeminent country in the world for science and research breakthroughs. And they're starting to get savvy. If the positions down here are decreasing, let's go bring the scientists over here and create institutes. And so that may be a result. But yeah, that doesn't serve the U.S. particularly well at all.
Right.
Yeah.
Right.
So what about one last question? And Jeanne, I think this is more probably, the grants, those are the graduate students and the postdocs, like these indirect costs. I think people have probably heard about that in the news. Can you tell us what that is? And would that affect like your program probably more?
It could. This one gets kind of complicated pretty quickly, but the indirect cost rates are rates that the institutions negotiate with federal funding agencies and for NIH, the National Institutes of Health, which is the largest funding entity or in the government. So those costs are intended to cover the things that the environment that needs to be present for the research to take place. So the buildings, maintenance of the buildings, the lights.
These are electricians, plumbers,
all those
painters,
things.
Air conditioning.
Air conditioning, carpenters.
And typically these rates are around 50% of the direct cost. So if the grant costs $100,000, when you write the grant, you're writing for $100,000 to do the research, and then you get your indirect cost rate. If it's 50%, $50,000, to help cover those costs
on top of the
on
100.
top of the 100.
Right.
So what they're saying is rather than a 50%, some institutions go as high as 75, 80%. The NIH and the government's only going to give 15%.
Oh, my gosh.
Yeah,
That's
a
huge.
huge cut.
You don't need air conditioning.
Exactly.
Yeah.
No,
Or
don't
lights.
need lights.
Or lights.
Can you do your work by candlelight?
Candlelight.
Yeah.
The good old days.
So
yeah.
romance,
Ron-
romantic science.
And
It was good enough for Fleming.
That's right.
And the variability from institution to institution, as Jeanne said, they're individually negotiated with NIH. But you could imagine if you're at an institution in New York City, Columbia, NYU, the cost for the buildings
Oh, my
is
Lord.
much
It's
greater
crazy.
than it
Yeah.
would be for
Yeah,
someone
we think
else.
rent in Nashville is bad.
Yeah.
Right.
You
Yeah.
know.
Yeah.
So again, if it went down to 15%, that means the institutions have to look for other dollars to keep the lights on, to keep the HVAC, the areas cooled. And so that means, again, fewer dollars for salaries, for other things that drive the research engine. So really scary, dramatic impact potentially on.
There is one thing I need to mention. I don't know what the thinking process was with the current administration when they decided to do this, but one of the potential reasons was that they wanted the institutions to spend more of their development money that they have available. And the money that the institutions have to spend that was given by donors is given for specific reasons.
Right. Yeah.
They want a library for children, or they wanted this. It's not a giant bucket of money that they can spend on whatever it is they want to do. So going and using
Finding
those
other
institutional
sources
funds
of funding to cover those costs is
is
a challenge.
is very
Yeah.
difficult.
Thank you guys for explaining that. That clears to me. That adds a lot of clarity to what we are hearing in the news and helping understand what's going on. I really appreciate
I often wondered about indirect costs until because it didn't mean anything to me when I was looking at it until I got it explained to me. So thank you. And thank you for this Absolutely fascinating. And thank you for making it so that I could understand it. I appreciate this so much.
Me too.
Yeah.
It was great to have you guys here
Thank
today.
you
It was
for
a
having
pleasure.
us.
Thank
Yeah,
you.
absolutely.
We will be putting information and links to some of the things that you have talked about on the website and in our socials. And we will be shooting that out to everybody here in podcast land.
Yeah, and
afterwards.
one of our goals is to kind of have an action item at the end of
Yes.
these pods for people to do, and if they get excited about something they hear about, they can kind of plug into it. And we're going to have lots of links to organizations where you can go if you want to learn more about biomedical research, learn how to support biomedical research,
And how to call your congressperson.
you need to call your congressman or woman, congressperson,
congresspeople.
and probably have some links. I think one of the things that I've been thinking about as we in this era of this decreased funding, it's like, okay, my mom has dementia. I want to support that. So private foundations, Ron, I don't know if you've gotten much funding from private foundations, if that might be something in this new era that becomes more of a funding source.
I do have money from private foundations. The problem is the amount of money that's available in that private system is so much less than is provided by the federal government. There's just not enough money to go around.
And you when you think of, well, that's the federal government is spending my tax dollars on this. Well, this old lady right here just had a couple of blood tests, which didn't even exist about 30 years ago. And I found out some very interesting information, and I'm acting on it, and it is not going to affect my longevity or my lifestyle. But I found out about it thanks to biomedical research and to your mice. Thank you very, very much. And thank you, taxpayers.
Yes.
Yes. And thank you, Ron. Thank you, Jeanne. I am
Thank you, Sue.
talk about over the moon. I am over the moon that you guys came and spent some time on our audio front porch.
It was fun. Thank you for giving us the
right.
opportunity.
Thank you very much.
All right, Sue.
All right. Pardon me. Let me consult my show notes, which are always impeccably arranged.
I just want to let everybody know, it is spring.
It is.
And everything's green. And if you like to get outside, this is the time of the year to go look for antler sheds. Like deer shed their antlers right now. And if you're into that, you got a couple more weeks before the grass gets tall and you can't see them. And right now it's perfect. I found 2 the other day on my walk with the dog.
And we're going to,
so awesome.
I'm going to post that picture of you with the antlers on.
Hey.
All right. You
Sounds
think we're
great.
kidding about being out here in rural the band. No, we are not. Our next show, okay. Ain't nobody here but us chickens. This show is called
I
What
thought you
Came
weren't
First?
singing anymore. Actually, there's a high demand for your singing.
And you're going to hear more of it on the next show.
But there was also a request from Mark to give you some singing lessons.
Thank you. I'd like to thank all of the feedback we've been getting. The show is called What Came First, the Chicken or the Egg Shortage? And we're going to talk about backyard chickens and what, the joys and perils.
There'll be some science, too, because we probably need to talk about avian influenza.
Yes. Well, we lead off with that. And
So we can then maybe we'll go to a more positive note. We'll start with that.
Okay. So let's wrap it up here. Thank you all for joining us. Thank you to science. Thank you, science. Thank you, Ron. Thank you, Jeanne. Thank you, everybody out there. I've got my glass here.
I've got mine.
Oh, but first of all, we got to tell them frontporchaf.com or wherever you get your pods.
Wherever you get your podcast.
Keep up with us on Facebook, Instagram, but
Blue
Sky,
but never
Twitter. Until next time. When
Cheers.
we gather on our audio front porch, grab a drink and solve all the world's problems.
All right.
Cheers.